developed mice that could actually form clear cell sarcomas genetically. So these were not tumors that we took out of a human patient and then grew in a mouse, but these were mice actually specifically growing a clear cell sarcoma because of the way that we modified their genes. So it's interesting, but initially the observations that were made in clear cell sarcoma identified the presence of this chromosomal translocation, or this fusion gene that was created by the chromosomal translocation, in all clear cell sarcomas. It's pretty easy to make the sort of leap to say, okay, well that must mean that is what is driving clear cell sarcoma. But we didn't really know that from there. What we knew was that they were always associated. Sometimes associations are not an actual causal relationship. And so what we did in the mouse was we tested this causal relationship, in that we took out of a human tumor only one thing, and that was just this fusion gene, this EWSR1 ATF1 fusion gene that had been associated with clear cell sarcoma. And we took that fusion gene and put it into a mouse and found that we could turn that gene on in the mouse, and what would happen was a full-blown clear cell sarcoma. It was uncanny. The tumors that developed in these mice were not differentiatable from human tumors. We took some of these tumors that happened in the mouse and we gave them to a pathologist who is a very good sarcoma pathologist, and we had this pathologist look at the tumors from the mice next to tumors from humans that were clear cell sarcomas, and they couldn't tell the difference. So this was really my first deep dive into the biology of clear cell sarcoma, was generating this mouse. We're using this mouse that forms clear cell sarcomas to try to understand not only the molecular drivers of the tumor, but also what are its unique vulnerabilities. So what are the chinks in its armor that we might be able to use to attack it with a chemotherapy or another kind of treatment. So we're using these mice not only as a platform to test treatments, but as another platform to try to dig into the biology of these tumors, partly because the tumors are so rare. Because the tumors are so rare in humans, we have a hard time getting lots of fresh tissue from human clear cell sarcomas. We can make the same kinds of tumors happen in the mouse, and therefore we can study them and study their unique biology, and then we correlate that biology to what we find in the human specimens, and then we can begin to sort of circle around or triangulate until we get to what is really the core of biology of this tumor.